are terminally differentiated, so these cannot regenerate, while all the cells of the body come from the interstitial stem cell. Now, what is interesting is people would like to know which regulatory events, signal pathways control transformation of a stem cell into differentiated cell. Now, before looking at this slide, please remember the slide I showed you for the hematopoietic stem cell. From the stem cell to become a blue cell, from blue to become yellow or violet, and so on, it needs specific instructions so that it changes its gene expression program. So it is important to know what regulates this development. So what is shown here is the multipotent stem cell that I mentioned, the interstitial stem cell. These arrows show that they divide continuously. But what is shown here is that they can differentiate once again into nematocyte, nerve cell, and gland cell. And if need be, they can form the egg and the sperm also. So people would like to know what decides whether a cell will become a nematocyte or a sperm or a neuron or so on. Okay, so these things are being studied in Hydra. Then how the stem cells of Hydra are different than stem cells of higher organisms or more complex organisms like us. Are there any differences? Are Hydra stem cells similar to us, our stem cells, or they are different? What makes them different? Why are our stem cells become different? Why have we lost the capacity of regeneration? These questions can be answered if you know details of the stem cells. Okay. Now I will come to another peculiarity of Hydra, and that is its capacity of regeneration. So this is an experiment carried out in our lab. What you see here is an entire Hydra on the left-hand side. And what we have done is we have taken a blade, and it's quite simple to do that. Once you're used to it, you can do hundreds of them. You take a Hydra, put it on a slide like this, and cut it with a sharp blade. Any blade will do. So what the student has done in this case, he has cut the Hydra into 3 pieces. Okay, so you get a middle piece, you get a top piece which has the head and with tentacles and mouth, and you get a bottom piece which has only the foot. What you see here is that over about 3 days, the top piece will regenerate the lost foot and the bottom piece will regenerate the lost head. So it shows a capacity of regeneration. Interestingly, the middle piece, which does not have either head or foot, can also regenerate both these structures, though it takes a little longer. So this happens in about 3 days. This takes about 4.5 to 5 days. Okay, but still only this piece can actually regenerate both the lost head and the lost foot and become an entire organism, which is Hydra. So in the lab, you can actually increase the number of Hydra by going on cutting them. Okay, this kind of regeneration is also seen in some other organisms, but Hydra regeneration is quite spectacular. You see regeneration in planaria also. You can make 256 pieces of a single planarian, and all of them will regenerate into a new planaria. Now let me tell you some details of Hydra regeneration. So I will go to the next slide, which shows it in a diagrammatic fashion. So this is the first figure. Concentrate on A. What is done here? You take one Hydra, cut it in the middle, you get an upper piece, you get a lower piece. You will see here that in some time, the lost foot is regenerated, which is shown in black, and the lost head is regenerated, which is also shown in black. So the regenerated tissue is shown in black. What I would like you to notice, I hope you have noticed that the regenerated Hydra are smaller than the original Hydra. So what is done here is that this Hydra has not grown in size. This piece has not grown in size, but it has only regenerated the lost structure. And here, this piece again has not grown in size, but it has regenerated the lost structure. That means the cells here or the cells here at the cut portion, which were actually forming the middle of the body column, now change their fate, change their structure, and instead of remaining cells from the middle of the body column, some of them form foot because there is no foot here, and the neighbors can form head because there is no head here. So the cells actually change their developmental pathway and form structures which are necessary for the Hydra to survive. So this is very important, that there is no growth to begin with, but there is only reorganization and differentiation of cells. So in animals, there are two kinds of regeneration. This regeneration in Hydra is known as morphallaxis, wherein there is no growth. So the cells here in the cut pieces are not dividing, but they only reorganize and form the lost structures. And there is another kind of regeneration in animals, which you see in, for example, in reptiles and amphibians. When you cut their tail or limb bud, you can see that. So let me explain this other kind of regeneration. Now imagine I'm a frog. Okay, I'm not, but I am a frog. If you cut my limb here, okay, and this has to regenerate, which happens in frogs and reptiles. So what happens is, at this cut piece, cells here will undergo what is known as de-differentiation. So they can actually now go into proliferation or mitosis. So cells undergo proliferation, a large number of cells are formed, and then they will form the lost structure. So to establish the normal original pattern, new cells will have to be formed. And this kind of regeneration is known as epimorphosis. Now, if you compare it with Hydra here, you can see that there is no such cell division to begin with. It only reorganizes the existing cells and they form the lost structures. So there are two kinds of regeneration. Hydra regeneration obviously is very quick, very fast, because it does not need new cells. Now, another aspect of Hydra regeneration, come to part B of the figure here, is that if you cut the Hydra into three pieces, the experiment I showed you in the last slide, then this middle piece, I already told you, can regenerate the lost head and the lost foot. So you can see this is the regenerated Hydra. What you must remember, and people have found out over the last 200-300 years, is that whatever you do with this piece, you can turn it upside down, you can centrifuge it, you can do whatever. Still, the new head, the regenerated head, will form always on the side where the original head was present. Okay, it will never form on the side where the foot was present. So head will regenerate on the side where original head was present, and foot will regenerate on the side where the original foot was present. And this has provided us with a very important mechanism in development of all organisms, all animals, including us. What is the mechanism? The mechanism is, why would these cells form the head and not the foot? And why would these cells form the foot but not the head? That is because in this piece of Hydra, there is some information available in the form of chemicals. So what is the information? The cells which are closer to the original head have the chemical for head formation in more quantity. So there is a gradient of molecules. The head forming molecule is highest in the top part and lowest in the lower part. So wherever you cut the Hydra, suppose this is the Hydra, if you cut it here, since this part is near, these cells will form head, not these cells. So there is an information there, and this is known as gradient of morphogens. This is a very important discovery that was made a long time back by a scientist called, many scientists, but it was put into a theory by a scientist called Louis Wolpert, and he only died 10 days back at the age of 91. He has made tremendous contribution to developmental biology by explaining how gradients are formed and how, because of that, proper structures are formed in our body at proper locations. Otherwise, my brain would have been somewhere in my knee. It is not there, I can assure you. But, otherwise, if development goes wrong, this can happen. The other thing about what Wolpert proposed is that once a head is formed, then there is another chemical secreted by the head, which takes care that second head is not formed. So it is very important that Hydra has only one head. So as soon as a head is formed, second head cannot form. This is also important, and that is why we have 2 hands, 2 legs, 10 fingers. If anything goes wrong in development, then some people will come with 6 fingers on one hand. Okay, but this is because of disturbance in the signaling molecules. So probably something went wrong when Ravan was developing, and he developed 10 heads instead of 1. So many mythological figures, when you see, you will see that there are more structures than are usually present, which can be explained with morphogen gradients. Okay, let me take the figure D now, C now, sorry. You can take a longitudinal cut in Hydra and that also regenerates. There is not much to describe here, but D is interesting. You can take a very tiny piece of Hydra, small piece of Hydra, and that will also regenerate into a small Hydra. So you can see that this organism has a fantastic capacity of regeneration. Okay, not only a small piece. Now, what people have done recently is that they have taken Hydra, separated all the cells, and they have centrifuged the cells. So you get a pellet of cells. Even that pellet can form an entire Hydra. Okay, so it has been shown that a pellet which is made up of only 100 cells. Please remember that the entire Hydra is made up of 100,000 cells, but even a 100-cell pellet can form a new Hydra. So Hydra has a tremendous capacity of regeneration. Now, when you study formation of regenerated hair, regenerated food, how small Hydra are formed, every time you can study these mechanisms at the animal level, morphological level, cellular level, molecular level, and so on. And therefore, obviously, developmental biologists find this model very useful to study how pattern formation is brought about. What are the regulators at pattern formation, whether at cellular level or at the molecular level? What kind of signals are necessary for this part to form head rather than foot, and this part to form foot rather than head? So all these one can learn from Hydra, and therefore, not surprisingly, Hydra is a popular model with developmental biologists. So let me just put things in perspective as far as regeneration in Hydra is concerned. So you see regeneration in Hydra, if you cut it into two pieces, the lower piece will regenerate the head. It will undergo only two steps, very simple and quick regeneration. First is wound healing. That's obvious. Whenever you cut something, first the wound has to heal, otherwise there will be infection. Okay, so wound healing takes place, which is followed by differentiation of cells. What do I mean by differentiation? The cells here, which were actually in the middle of the body column, now change their fate and they become head. So that is differentiation. So Hydra is a very simple regeneration, wound healing followed by differentiation. If you go to planaria, then it is a little more complex, wherein you actually have wound healing. That is, of course, the first step which is necessary, followed by a structure called blastema. It's a after wound healing, it's a specialized structure which leads to differentiation of local cells into formation of the lost structures. If you come to vertebrates, which I described a while ago, limb regeneration, let's say in frog, then you have more complex regeneration. Firstly, the wound healing takes place, then the cells near the cut de-differentiate. Why do they de-differentiate? Because unless they de-differentiate, they cannot divide. Differentiated cells in our body cannot divide. If cells start dividing uncontrollably, it causes cancer, as you know, in simple terms, tumor, not cancer necessarily, but tumors. So wound healing is followed by de-differentiation, which is followed by blastema formation, and finally differentiation, so that the original limb is grown again. The lost parts are grown again. So as you go from more simple to more complex organisms, the regeneration mechanisms also become more and more complex. And if you come to us human beings, you know that we cannot regenerate. If you cut my hand, I won't be able to regenerate. Good, it's a virtual workshop so that you can't do it. But we can regenerate our liver to some extent, maybe pancreas to a little extent. We shed our skin every day, but most of our organs have lost the capacity of regeneration. And people would obviously like to know what is so peculiar about organisms like, let's say, Hydra, starfish, planaria, which can regenerate, who can do it day in and day out, and whether some tricks from such organisms can be applied, at least in culture, to grow organs in vitro. So that is the importance of this study. So there are two things. When a person like me studies Hydra, there are two reasons why people like us study Hydra. Firstly, it is natural scientific curiosity as to how a particular organism survives, develops, reproduces, and so on. The second is whether knowledge from such organisms can be applied for something useful in human beings, either to make our lives better or to avoid diseases. Okay, Now I'm coming to the last couple of aspects of my talk. I am going to finish in maybe another 10 to 15 minutes, which will leave some time for discussion. Okay, so this is another experiment that developmental biologists do in different organisms. And this is an experiment done in Hydra. Please see the date, 1909. This lady, Ethel Brown, what she did was she took Hydra. By the way, Hydra is just about a centimeter and a half long. So it's a tiny organism. You can see it with naked eyes, but if you want to see it properly, you have to have binoculars. Anyway, so what Ethel Brown did was she took a piece of Hydra from one Hydra, a small piece near the hypostome or the conical structure which has the mouth, and transplanted it or put it into the body column of another Hydra. And she found that it leads to growth of another Hydra. Okay, that means I'll just explain it briefly. So when you take cells from the hypostome of this Hydra and put them here, the cells here do not anymore remain the body cells, body column cells, but they actually form a new head. So they form a new axis. That means the black cells, the black group of cells that are transplanted here, secrete some signal so that the surrounding cells now change their fate, now change their structure, and instead of remaining body column cells, they form new body column, a new axis. So if you take a piece, again, the same experiment is shown here at the top, but if you take a small piece from the foot, again you get a new axis, but this time the foot comes out first. In this case, the head came out first. That is because cells are from the head and here the cells are from the foot. If you put the two cells together, then they neutralize each other. There is titrating out and you don't really see any effect. That means these cells have one kind of signals, the head forming signal. These have the foot forming signal. If you put them together, then neither the head nor the foot is formed. But if you put them alone, then they influence the surrounding cells and new structures are formed. Another interesting experiment in Hydra, which is quite interesting, and that's why I am explaining it. So if you take this piece again, small piece, and transplant it on a near the existing head, okay? So now in this case, the recipient is, the transplant in the recipient is near the head, you don't get any effect. And it is believed that because the existing head does not allow development of another head. That's why we have only one head, because this mechanism is active. This mechanism was probably lost in case of Ravana, who got 10 heads. Okay, but if you do this experiment in a different way, so now you take a transplant, put it on the recipient after its original head is cut. So cut the head and then put the transplant, then you get a new head here. And in the meantime, the original head also regenerates, because you know that Hydra can regenerate. So now you can get a two-headed Hydra, but not unless you cut the original head. That shows that there are molecules in Hydra head which do not allow more heads to form. Now, please remember that these are very important insights into how structures develop. So from a simple, obviously you cannot do such experiments on higher organisms, especially human embryos. You are not supposed to work with them. And therefore, knowledge gained from other organisms tells us how our development can possibly take place. So this is very important experiment and is very useful. Similar experiments were done in frog embryos a little later after Ethel Brown's experiment, wherein a piece of frog embryo from the dorsal side, if you put it on the ventral side of the recipient or the host, you get a twin embryo. That means these cells can also send signals so that this forms another embryo. This work in frog got a Nobel Prize in Medicine in 1928. But what I would like you to understand here is frog is a vertebrate, which is very similar to us. It has these structures which are known as organizers because they organize a new tissue. But even an evolutionarily ancient organism, which arose millions of years ago, also has a similar mechanism for development and pattern formation. So it shows that we have all evolved from common ancestors through mechanisms which are very similar. Okay, now the last aspect that I would like to convey to you is that I told you that there are organizers in Hydra and there are organizers in humans. So there are many similarities in how pattern formation happens in Hydra or even in humans. And why this is so is because now people have found out that a large number of genes which code for various proteins in Hydra are very, very similar, remarkably similar to genes found in the human beings. So even our own work has shown that many of the transcription factors, transcription factors are the ones which are responsible for initiating gene expression, or the cell signaling molecules which signal cells, neighboring cells to become either a liver cell or a pancreatic cell. So these cell signaling molecules or DNA repair proteins. Our DNA is damaged continuously because of various reasons. It is repaired all the time. So DNA repair proteins, transcription factors, cell signaling molecules from Hydra show tremendous amount of similarity, both at the structural level and many a times at the functional level, with molecules from human beings. This shows that the basic toolkit, as they say, you need a toolkit to build a machine. So if you need a toolkit, a genetic toolkit to build an organism, so the basic toolkit of genes to build a multicellular organism evolved millions of years ago. And that is being used again and again and again and again to make different kinds of animals. The reason behind making different kinds of animals, as you know, is because the genes are expressed at different times and at different locations, and therefore we end up making a different structure. I don't have a picture right in this presentation, but let me quickly explain what I mean by this. So if you look at a chicken foot, it will have fingers. If you look at a duck foot, it will have waves in between because it needs them for swimming. Okay, but both birds and both chick and duck are birds, so their genes are likely to be very, very similar. In fact, they are very similar. However, what happens in case of duck, the webbing in between the fingers remains forever in its lifetime, while in case of chick, this undergoes what is known as programmed cell death and fingers are formed. The same mechanism is also used when our fingers are formed. Actually, these are all connected to each other in our early development, but the tissue in between undergoes programmed cell death and we get 5 fingers. If that goes wrong, sometimes some people get 6 fingers. Okay, so what I'm trying to tell you is that with the same kind of genes, you can make different structures. So the genetic toolkit evolved only once in evolution, and it is responsible for all the biodiversity that you see around. This aspect I'm going to discuss in a little more detail in tomorrow's talk, so I will leave it at that. So various proteins sometimes perform the same function in diverse organisms or different functions. Let me quickly give one example. We have a molecule in our body which is known as vascular endothelial growth factor. It is a growth factor. It is also responsible for development of new blood vessels in our body and even neuronal connections. Okay. Now, Hydra does not have blood because blood is a mesodermal tissue. Hydra does not have mesoderm. I told you that it's a diploblast. It has only ectoderm and endoderm. It does not have blood, but it still has VEGF. And what we have found ourselves in our lab is that this VEGF is not responsible for blood formation there, but it is responsible for branching of the tentacles. So what we see here during evolution is that some proteins carry out different functions in different organisms during evolution. So VEGF originally evolved for branching of tentacles, but later on recruited to form blood vessels in higher organisms. So this happens that many a times they are recruited to perform either additional or entirely different functions in different animals. We have now people have now been able to form transgenic Hydra. See, the problem with Hydra is that you don't get too many mutants, because I told you that the cells are continuously being renewed, and therefore it is very difficult to get stable mutants. See, for getting a mutation, the mutation needs to be fixed. Now, if a cell has a mutation in Hydra, and if the cell is going to be thrown out of the body in 20 days, obviously it will not be a stable mutation. So it's very difficult to get either transgenic Hydra or stable mutations. And therefore, this model suffers from this problem that it's not very easy to work. So as compared to, let's say, Drosophila or zebrafish or Sinorhabditis elegans, it's a difficult organism to work with. However, because of its tremendous capacity of regeneration, because of its capacity to remain young forever and refusal to die, it is still a very interesting biological organism. Please remember that when you choose an organism as a model system to answer the questions that you are asking, every model system will have certain advantages and certain disadvantages. So you'll have to choose the organism that you choose very carefully so that it can answer the questions that you are asking. Every organism is different. So ideally, a model organism is a good model only for itself. But then you cannot work with animals, so you have to work with other mammals. So you have to make a trade-off and then use model organisms to understand basics processes in animals. So very briefly, we carry out many different things in the lab. I'm not going to go into details, but we have been doing it since the year 2000. By the way, it will interest you to know that in the year 2000, I brought one hydra, single hydra, from a pond in botany department of Pune University. And all the thousands and thousands of hydra that I have in the lab and I have used over the last 20 years are all progeny of the same hydra. So I have collected one hydra in the year 2000 and I am using only progeny of that hydra. So it's a clonal culture going on for the last 20 years in the lab. Because hydra is a good organism to teach biology, and we have been supplying it, and it is actually in the syllabus of Bombay and Pune University, how to study hydra regeneration. We were studying it to many colleges and universities, even outside Maharashtra. Now we have come out with a hydra kit. It's not very expensive. The idea is to help all teaching institutions to use hydra for teaching. So we provide some hydra, we provide its food and so on, and it can be ordered from Agarkar Research Institute. So this is our contribution to bringing hydra as a model system for teaching and research in this country. And recently I wrote an article for the journal Resonance, which is published by the Indian Academy of Sciences in Bangalore in September 2020. This is a review article which can be used by beginners, all by students, to understand this model. Actually, it will have all the details that I talked today, but only thing is you can read it at leisure. This is freely available. You can download it freely and you can see this article as a beginning. Later on, you can move to more complex organisms. So our endeavor has been to popularize hydra as a model system for teaching in India, and we use it ourselves for research in biology. So these are many of the students. All that I talked about has been the work has been carried out by students. I only talk, they are the ones who work, and it's funded by many organizations. So what I'll do here is probably, I'll stop. It is 11:53 and leave some time for questions.
I request all the participants to unmute themselves because the session is open for queries.
Don't be shy. You should ask questions. Otherwise, I will not let you go for lunch. Okay. I hope you like it. They can ask me by email also. They can ask me tomorrow also. But we'll wait for some time. People will probably have the.
Okay, sir. Okay, sir.
Good morning, sir.
Good morning.
Myself, Madhuri of MBZC, final year, Nizamabad College.
Yes.
As you explained about regeneration of hydra, can we do it in our normal laboratories?
Absolutely. Absolutely. I know some people who keep hydra as pets also. But one thing, you have to be careful because hydra feeds only on live food. So you have to provide it with food twice or thrice a week, and then clean. So you have to keep it like a pet. As you keep a dog or a cat and take care of that, you have to keep Hydra. You can do regeneration experiments or budding experiments in Hydra in any lab. I have actually taken Hydra to schools, cut the Hydra in schools, and they have seen regeneration after 3 to 4 days. So you can do it anywhere. And that's why I'm saying that it's a useful model, because animal ethics problems do not come. You can still show, and you can yourself see how Hydra regenerate over the period of 3 to 4 days. It's quite easy. Have I answered your question?
Good morning, sir. I'm Aparna, lecturer in microbiology from Social Welfare Residential Degree College for Women, Mehboob Nagar. My question is, for how long, how many times the Hydra will regenerate?
Well, indefinite. Nobody has seen a Hydra stopping regeneration. In fact, I told you that it never stops. We don't know. Nobody has seen a Hydra dying. It remains forever.
Okay, so we can tell it as immortal.
It is considered to be immortal. And since you come from microbiology department, let me mention quickly that Hydra also have its own microbiome, okay, like we have.
Okay.
And it's very important, because if you change the microbiome, it sometimes affects budding. So, you know, our association with microbes, as you know, we have more microbes than our body cells. Hydra also has microbes of different.
Thank you, sir. Thank you so much.
Okay.
Yes, sir.
Any other questions?
Morning, sir. This is Sarita from LB Nagar College.
Yes.
I'm a faculty of zoology.
Okay, Sarita.
Sir, I have a small clarification, sir.
Sure.
Is this regeneration under the neural control of Hydra? Because we have a nerve network in Hydra.
Yes, but it is not clearly known, but it is more under the control of the local chemicals rather than neurons. Okay, so okay. So I told you about those morphogens, those signals
Yes,
that
sir.
are present. So
Yes,
those
sir,
are the ones which sort of instruct the cells around to either form a head or a foot or whatever is lost. So there is no neuronal component immediately involved there. It's not necessary also. Okay.
Hello, good morning, sir.
Good morning, Sana. Sana
Yes, sir.
Fatima. Yeah, go ahead.
Yes, sir. Sir, I want to understand, can we able to understand through next generation sequencing why Hydra is immortal?
What is next generation sequencing will tell you only. So Hydra genome is already sequenced.
Then why we haven't understand why it is immortal so far?
Let's say why we haven't understood how we change our moods so frequently.
Sir, you are not audible, sir.
I've switched on to another this thing. Now I will speak again. Okay. Can you hear me now?
Yes, sir, you are audible now.
Okay. So I have a power failure. I switched on to some other hotspot. So, by just looking at the sequence, we do not know many things. So likewise in Hydra also, we have no idea why it generates. People are trying to Have I answered your question?
Yes, sir.
Okay. So there are many things we still don't know, although we know what kind of genes are present even in human beings. And it's very complex. Genes don't explain exactly everything. Hormones also we do not understand properly. So there are many things yet to be done. And that is precisely the reason why we want young people like you to come into biology. Because biology seems to be the last frontier now, wherein there are so many things to be learned.
I request the other participant to mute yourself.
Actually, today I did an experiment. Instead of sitting down, I put a big table and I'm standing and moving around. So it is a little virtual talk. If I was there, I would have done some more, acting and shown you. But anyway, we can't do anything about it. Maybe next year.
Okay, sir. We expect you in live after this pandemic situation, sir. Sir, this is
Absolutely. I
Dr.
have come to Telangana many times with Dr. Munierkar. We can come again. Okay, go ahead. I'm
Okay,
through.
sir. Okay, sir. Thank you, sir. This is Dr. Sushma from TSWREIS in Nizamabad, lecturer in chemistry. It's a fascinating lecture, sir. I have ever interested in this Hydra, sir. And you have explained a Nidarian organism called Hydra with some interesting factors, sir. It is having neurons which can help in its active feeding. And right from the introduction of Tremblay, you have shown us the structure and cellular dynamics of that organism. And you explained an interesting factor that it is an organism which escapes aging and called it as a potentially immortal organism which refuses to die with the help of stem cells and how they regenerate Hydra in different portions when it is cut. And thank you so much, sir, for enlightening us over the knowledge of Hydra. Thank you so much, sir. We are pleasure to have you, sir. Thank you.
Thank you very much. So I'll leave now. Your next is by Dr. Susan. Okay, thank you.
Thank you, sir. Thank you. Thank you so much, sir, for the.
Thank you so much, sir.
Thank you. Thank you. I also enjoyed the interaction very much. The coming speakers are also excellent participants to take benefit of them. Okay.
Yes, sir.
Okay.
Thank you, sir.
Thank you, sir.
Now I request Ms. K. Anita, lecturer in chemistry, to introduce our next speaker, Dr. Susan Apel. It's a great privilege for me to introduce Dr. Susan Ipan. Stanford University has recently included Dr. Susan Ipan in the world 2% top ranking scientist in the category plant biology and botany. A 15th batch BART training school scientist in biology and radiobiology, she retired in the year 2011. Currently, she is engaged in motivating scientific temper among faculty and students at Union Christian College, Alua, as an adjunct professor in biosciences group. She is considered one of the pioneers in transgenic plant research, and her interest varies from plant biotechnology, production of secondary metabolites from in vitro cultures, and phytoremediation. She has published 140 research papers, contributed several book chapters, and presented papers in several national and international symposia. Her latest paper is on phytoextraction of uranium using vetiver plants. During an Indo-FRG bilateral program in 1986 at Free University Berlin, she developed the first transgenic grain legume from protoplast using 3 different techniques. Later, she developed transgenic plants which could take up heavy metals and degrade organic pollutants. She was also involved in the Cotton Mini Mission project of Government of India and was a collaborator for several BRNS-funded programs all over India. Scientists from Russia, Cuba, and Syria have come to her lab to get trained in latest techniques. She had been in the editorial board of international journals In Vitro Plant and BMC Biotechnology. She received Elsevier Award in 2010 for the Best Quoted Paper of the Year. She is also a recipient of UNESCO and British Council Fellowships. We are glad to have you here, ma'am. I request Dr. Susan Ipan to take over the session. Thank you.
Am I audible? Hello? Hello?
Yes,
Can you hear?
Yes, ma'am.
Can you see the slide? Can you see the slide?
No.
No, ma'am.
Cannot see. 1 minute. Just a moment.
Okay.
Share. Share. Can you see now?
Yes, ma'am. Yes, ma'am.
Is it okay?
Yes, ma'am. It's clear.
Can I start?
Yes, ma'am. You can start.
Good afternoon, everybody. Dear participants and our prestigious speaker, Dr. Surendra Gaskardi, then Dr. Snehalada, Dr. Khan, and all the other distinguished speakers like Rita Mulekar and other participants. It's a pleasure for me to deliver a talk on crop improvement using biotechnological applications. Before I go to the actual talk, I would like to talk a few things about the journey of biotechnology and the journey of plant breeding so that you get familiar with the talk. So biotechnology, as you know, has been practiced from time immemorial. Like you know that cheese, wine, etc. have been made many centuries back. This is winemaking. But now the term biotechnology is a new term coined in 1919 by Karl Erecki, a Hungarian engineer who developed bioproducts rapidly using biological materials. Currently, biotechnology is used to improve human health, environment, and to produce new plants like this tomato, which can be kept for 2-3 months without spoilage. You can produce vaccines, you can produce pharmaceuticals, other novel foods, agricultural products, etc. to help the society. So now coming to plant breeding started when man started domesticating plants for food. This must have happened at least 10,000 years back when plants were selected and bred for giving us proper food. Around 700 BC, Babylonians and Assyrians artificially pollinated date palms. It was only in 1717, Mr. Fairchild, he produced a hybrid between two varieties of carnation. The first artificial hybrid in dianthus or in carnation. Later, Joseph Kohlreuter, a German scientist, crossed a tobacco plant with a solanum plant or a potato plant, and he produced a hybrid. So between two different genus were used for crossing, and he produced a hybrid plant. In 1857, another person called Bill Morin, he started selecting plants individually, and then he bred them for the next generation. Now you are all familiar with the milestones in biotechnology, especially Mendel's laws of inheritance, where he used pea plants. As you know, Gregor Mendel lived in Brunn, Austria, and now the place belongs to Czechoslovakia. Gregor Mendel was a priest or a monk, and he used to stay in a monastery. He was actually a mathematician and a statistician. He was very good in that, but he was always wondering how. Sorry. How Can you mute the mic? Whoever is speaking. Yeah, he was a monk in a monastery. And he wanted to work on mice. And he kept many mice in his monastery. But all the other priests were against keeping mice in the monastery. So he switched over to pea plant. So in his monastery, there was a huge garden. And he collected several varieties of pea plants having purple flowers, pea plants which are tall, which are short, which are having flowers on the axil, which are having green color pods or inflated pods. He collected many plants and he bred them for several generations and maintained in his monastery. After that, he wanted to know how plants are characters are inherited from parents to offspring. He was always interested to know how dogs give birth to puppies, how human beings give birth to human babies. Why not the other way? So plants, how they give birth to their children and the next generation, and how characters are inherited from this generation to this generation to the next generation to the next generation. It was very much inherited. For his experiment, as you know, he first crossed a purple flowered pea plant with a white flowered pea plant, and you expect maybe both purple and white flower, but to his surprise, in the first generation he could note only purple flowers. So what happened to the white flowers? Where has the white character gone? Then he selfed the plant, and in the next generation he got a large number of plants with flowers, and when he collected the data and analyzed, they were in the ratio of 3:1. And he was a good mathematician and he was a good statistician, and he reached the conclusion that each character, that is purple flower color, is governed by two alleles. He did not know anything about genes at that time, and he said they are present in the plant as two distinct alleles, and alleles do separate, like in the next generation. So using different characters like seed shape, seed color, flower color, pod shape, pod color, then flower position, whether it's in the axil or in the top, and then height of the plant, tall plant, and crossing with a dwarf plant, etc. He reached the conclusion that physical traits are inherited as particles. He did not know what are those particles. He had no idea that it was DNA or gene or anything of that type. And he knew that the character is passed on from generation to generation, from parents to the offspring. And they remain as two in number. One comes from the father and one comes from the mother plant. And they remain as a double, and one character dominates over the other. When the character is like he represented as capital R, when both are dominant, and capital R, small r, when one is dominant, other is recessive, and both small r, when both are recessive. When I look at the other milestones in biotechnology, you see that in 1870, mitosis was discovered. Mitosis is different from meiosis, which take place in germ cells. As a result of mitosis, you get the same number of chromosomes. In 1873, Robert Koch had demonstrated staining of microorganisms. And in Mendel had published his laws in 1865, but nobody noticed the Mendel's laws of inheritance, and nobody gave any importance. But afterwards, after a long time, in 1900, from three laboratories, scientists rediscovered Mendel's laws, and they understood how important Mendel's laws are. Then by the time Walter Sutton found out that chromosomes occur in pairs, so Mendel had postulated that the factors remain as alleles. So that came true when he found that there are two chromosomes for each one, for two pairs of chromosomes, one pair of chromosomes for each. Then came the work of T.H. Morgan. He worked on fruit fly, that is the Drosophila melanogaster, and he worked out the genetics of that. Then the actual name gene was put forward by William Johansen in 1999. And 1914, lot of work was done in hybrid maize, and the hybrid maize, vigor or heterosis was demonstrated. So meanwhile, other developments in biotechnology was taking place. I'm not going into the details. You all would have studied all these things. Nobody had an idea what is a gene, what is DNA, nothing of that type at that time. Then at that time, Dr. Frederick Griffith and his group, they demonstrated a transforming principle. So there are two strains of a bacteria, the non-virulent strain, which is shown in blue color, and virulent strain, which is shown in red color. When you infect a mice or inject a mice with a non-virulent strain, the mice all survive. None of the mice will die. But when you inject a virulent strain into the mice will die. Then he did a third experiment. He heat killed the virulent strain and mixed with the non-virulent strain and injected into the mice. What happened? All the mice survived. None of them died. Then again, he heat killed the virulent strain and mixed with the non-virulent strain and injected into the mice, and all the mice died. That shows a molecule is getting transferred from the virulent strain to the non-virulent strain, making it virulent so that the mice dies. At that time, nobody knew it was DNA. And later, during the same period, you know about Alexander Fleming. He discovered penicillin. Beadle and Tatum, working on Neurospora crassa, they elucidated the principle of one gene will produce one enzyme, genotype, phenotype, etc. Animal cells were grown in vitro. Plants were shown to be totipotent, that each plant cell, when you culture them, whether it is a somatic cell, it can become an embryo and become a complete plant. And this was demonstrated in carrot. You take a cell from carrot, culture in a medium containing 2,4-D, which is an auxin, high auxin, remove the auxin and put it into flask liquid medium. Each cell will become an embryo, and they can be plated out. The typical stages of embryogenesis and plantlets can be produced. So you know about the structure of DNA was elucidated by Watson and Crick. I'm not going into the details. Then other milestones in biology, like the genetic code was cracked then by Severo Ochoa and Arthur Kornberg. I happened to visit them. You can see me here, visit the place. Then they synthesized infectious viral DNA, and they got Nobel Prize for biological synthesis of DNA and RNA. Other milestones in biotechnology include the discovery of restriction enzymes to cut DNA and also to paste them together using ligase. So this was made by Paul Berg in 1972. In 1978 to 82, human insulin gene was commercially produced. You know that humans who are having diabetes, many a time they need insulin. And how were they getting insulin before 1970s? They were getting insulin from pancreas, isolated from a slaughterhouse where cows and pigs and all were killed, and insulin was isolated. But because of the cloning techniques developed in 1970s, human insulin gene was commercially produced in a bacteria without the killing of animals. Okay, now in 1980, Cary Muller, he demonstrated polymerase chain reaction. He got a Nobel Prize in 1993. You all are now familiar with PCR, RT-PCR because of COVID. You all are familiar with that. In 1985, DNA fingerprinting. DNA fingerprinting is widely used to detect people who has done the crime at the crime site by doing DNA fingerprinting, or to find out who are the parents, who is the father of the child when there is a court case. Then in 1983 to 85, genetically engineered plants were produced. Then a recombinant vaccine was produced for the first time in tobacco. And in 1987, tomato with antisense RNA with a extended shelf life was produced. And 1990 first gene therapy. Dr. Rita is going to talk about this today, and she will also speak about how genome project started. And cloning of Dolly was done, a sheep was done in 1997 by using the egg cell and enucleating the egg cell and introducing a somatic nucleus into that. Then 1998 the embryonic stem cells were cultured and the stem cell therapy started. Now let me come to the plant tissue culture. In 1902, Haberland said that plant cells are totipotent. Starting from a single somatic cell, we can produce a complete plant. Like, just now our earlier speaker had said that Hydra can also regenerate. So plant cells have a potential to regenerate complete plant starting from a single cell, provided you culture them, nurture them, and provide them with the nutrients and cytokinins and auxins and other things. So in 1937, Philip White from USA for the first time cultured tomato roots in vitro. In 1957, Skoog and Miller, they found that when you change the ratio of plant hormones in vitro, that is auxin and cytokinin, it can determine the production of shoots and roots in vitro. And in 1965, our Indian scientists, Dr. Sipra Guha and Sadish Maheshwari, demonstrated anthiculture for production of haploids. Well, you know what is anthiculture? And the anthers contain microspore or pollen grains which are equivalent to sperms. So they contain only half the chromosomes. So production of haploids by a culture of microspores or anthers or pollen grain was demonstrated for the first time. And in England, during the time, Dr. E.C. Cooking, they said that plant cells are, that plant cells are different from animal cells. And one difference is plant cells have got big cell walls, So E.C. Cooking said that you can remove the cell walls by digestion with enzymes and you get naked plant cells, plant protoplasm, he called it. And this can be used for introduction of genes. This can be used for somatic cell hybridization. And in 1971, Takebe from Japan showed that regeneration of plants from protoplasm for the first time. In 1974, some chemicals like polyethylene glycol and calcium were added to the medium to induce fusion between two different cells to produce the somatic hybrids. In 1978, somaclonal variation was detected. Somaclonal variation, I will come to the details later. And the first transgenic tomato flavor saver was released into the market. And the human genome project started. Rita will be talking about it. It started in 1990 and completed in 2003. Then this started the molecular medicine, personalized medicine, etc. It is available, the information is available in the internet. DNA sequencing started in several organisms, right from bacteria, yeast, and the Cenobartis elegans, that is a worm, then a plant, very model plant called Arabidopsis thaliana, then Drosophila melanogaster in 2013. Jennifer Doudna and Emmanuel Charpentier received the Nobel Prize, and they demonstrated CRISPR-Cas9, for which they received the Nobel Prize in 2020. So this is a picture of Emmanuel Charpentier and Jennifer Doudna. They said that CRISPR-Cas9 can be used as molecular scissors for genome editing. I think Rita is going to talk about it. Now let me come to the main topic. There's a crop improvement for feeding the world. You know that world population is increasing alarmingly. That is, by 2050, we will reach 9.7 billion in the earth. So you need food security. That means access to sufficient quantity of affordable, not only affordable, it should be nutritious food. So we have to produce more food. and our land is decreasing. Our water levels are going down. Our climate is changing, and new diseases and pests are coming and attacking plants. Like the COVID has come, plants also are facing so much of problem from new diseases, pests, and all that. So what are our food crops? We have cereals and millets, wheat, rice, maize, pearl millet, finger millet, etc. Pulses like chickpea, pigeon pea, black gram, and green gram. And we Indians depend on these pulses for our protein intake. Then we have oil crops like sunflower, groundnut, soybean, Indian mustard, coconut, etc. Fruit crops, vegetables, sugar crops, spices, beverage crops, etc. You all know about all these plants. Now, why do we have to increase food production? These children staying in slums and in villages, they don't get enough food to eat. It's very important that children get enough food to eat in their formative years, because the brain develops in the very young age, maybe up to 7 to 8 years. So if they don't get food in the young age, and if they don't get nutritious food, the brain will not develop properly. So poverty and starvation, and people don't have job. Food may be available in the market, but they have no purchasing power, and malnutrition may be there, which will affect the growth of the country, because the brain development of children is very, very important. And not only that, about hidden hunger. What is hidden hunger? See, people go to McDonald's and other restaurants and eat junk food. So as a result, what happens? They fill their stomach, but they have hidden hunger. They lack some nutrients like iron, zinc, etc. That is known as hidden hunger. And not only that, global warming due to climate change leads to crop failure. Crops don't grow. When there is flooding, when there is high temperature, you don't get the yield. Sudden rain comes, shortage of water for cultivation, and increase in pests and diseases. Then what are the types of reproduction in plants? There are two types of reproduction, as you know, asexual reproduction and sexual reproduction. In asexual reproduction in plants are very resilient. That means they can propagate from underground stem, corms, tubers, bulbil, stem cutting, layering, grafting, apomixis, etc. In sexual reproduction, you see this flower as a female part, that is a gynoecium, and androecium, and androecium contains the pollen grain. The pollen grain has to fall on the stigma, and the male gamete has to go inside through the pollen tube and fertilize the egg cell. So this is a plant fertilization. Plant fertilization is very important to produce the seeds. Any change in the climate can stop plant fertilization. That means you don't get seeds, you don't get food to eat. So now, what are the methods of crop improvement? I am going to talk about the conventional methods as well as the latest methods. So one is introduction of plants. You introduce a plant, you don't have a plant of what you want in India. So you introduce a plant from Taiwan, you introduce a plant from Australia, then that is called as introduction of plants. Then second method is selection. So you go to a field, like in this field, suppose there is a crop attack of the crop by a particular disease like a rice blast. And among the plants, all the rice plants are dying, but one plant is surviving, and that is known as, that is selected, that is called as selection. And the third type of plant breeding is hybridization. You can cross one plant, high yielding plant, with a disease resistant plant. That is known as hybridization, and you get a good plant with a high yielding with the disease resistance. And hybridization is also used for heterosis breeding. You have heard about hybrid corn. The hybrid corn is very high yielding. Like a lot of yield is very high. This is produced due to hybridization. I will come to the details little later. Then there is polyploidy breeding. Many plants are vegetatively propagated. In such plants, polyploidy breeding is very good. Then you know that mutations are induced. Naturally, there are spontaneous mutations, but mutation frequency can be increased by mutation breeding, by exposing the tissues or the seeds to gamma rays or X-rays or to chemical mutagens. Then plants tissues can be grown in tissue culture, and somoclonal variation can be obtained. Then you can it is not possible, as you know, to cross rice plant with a wheat plant, but using somatic cell hybridization you can cross a rice protoplast with a wheat protoplast and you can produce somatic hybrids. Then, as I said before, and the culture can be used, that's microspore or pollen culture can be used to produce homozygous plants, and homozygous plants are very important to produce hybrids. So what will take many years to produce homozygous plants can be achieved in a single generation by culture or microspore culture. Then the latest techniques, that is after 1980s, then gene transfer using agrobacterium, particle gun, etc., to produce transgenic plants. All of you have heard about genetically modified plants. Then molecular markers can be used like RFLP, RAPD, AFLP can be used for selecting plants at an early stage, and the information can be used for plant breeding. And of course, the genomics information also can be used for plant improvement. And the other latest technique is genome editing. Now, what about introduction of plants? You are taking a genotype to a new place or environment where they are not grown previously. Previously, the plant is not grown in that area. For example, you bring a plant from Australia. For example, in introduction of International Rice Research Institute from Philippines, you can bring the rice varieties to India, or you can introduce wild sunflower plants varieties from Russia to India, or you can introduce Mexican beet, which are high yielding, to India. This is known as introduction. This is the early method of plant improvement, because we don't have the plant in India and we are bringing a plant from outside. You all have heard about Green Revolution. You must have wondered, what is Green Revolution? In 1960s, there was a severe shortage of food in India. For example, wheat production had gone down, rice production had gone down, and at that time, the M.S. Swaminathan was a director of Indian Agriculture Research Institute in Delhi, along with the then food minister, not doctor, C. Subramaniam. They decided to import some varieties of wheat from Mexico. And Dr. Norman Borlaug, he was a scientist from USA working on wheat in Mexico, and he developed some novel short varieties of wheat. So that wheat varieties Norin 10 was brought to India, and it crossed with our local varieties to produce Kalyan Sona, Sharbadi Sonora, etc., which are high yielding, which had a short stature, which don't fall down when a wind comes, and which are high yielding. Okay. Now, similarly, is farmer in Taiwan, among the tall rice plants, he found a dwarf rice plant that is known as DG Vojan. So this plant selected by this farmer in Taiwan was developed into IR8 and other varieties of rice, and they were introduced into India. So in 1960s, all the world nations were looking at India because we were a begging nation. We were standing with a begging bowl for the food. You know, like wheat had to be brought from USA and all that. And from this, because of the Green Revolution, because of the agricultural inputs like a lot of fertilizers, pesticides, etc. were used, and these varieties were imported, and people started improving our varieties. From a begging nation, we became a nation which was supplying wheat and rice to other countries. Now, how do you select plants with a desirable trait? I said that from the 16th century onward, selection was practiced in Belgium and other places. A dwarf rice variety called DG Vojan was selected by a farmer in Taiwan, and leading to high-yielding varieties called IR8 and other varieties, so which resulted in improving yield in our own India. So disease, suppose there is a disease, you can select for a disease-resistant plant. This is called as method selection. This selection method is used for improving the plants. You want a salt-tolerant variety, and you can grow the plants in presence of salt, And whatever varieties are surviving, we can select. This is salt tolerant varieties. This is plant hybridization. You can cross a tomato with a tomato, or with a lettuce with a lettuce, or a tobacco with a tobacco, one variety with another variety to improve characters of the particular variety. Or you can cross one species with another. For example, Nicotiana tobacco and Nicotiana silvestris can be crossed. Or you can have intergeneric hybrids. Wheat, you all are familiar with the bread wheat, that Triticum eastivum. It was crossed with rye or cereal cereal to produce triticale, which is a very hardy cereal. It has got 8 X chromosomes. And sometimes what you do is you hybridize the plant. For example, you have a high yielding plant, which is B, and you cross it, but it is susceptible to a particular disease. So you select another plant which is disease resistant, that is A. So you cross B with A and you get AB. But you want high yielding plant with only one character, that is disease resistant from this plant. So this hybrid, you go on back crossing with the high yielding B, maybe for 5 to 6 generation, and in each generation you select for yield as well as for disease resistance. So a variety with high yield and disease resistance is selected by hybridization and back crossing. So plant hybridization also can be used for improving hybrid vigor or for heterosis. See, 2 maize lines are here, and these are bred for several generations, to get a pure line. And then they are crossed and you get a hybrid with very high yield. So hybridization can be used to produce hybrid vigor. You are familiar with tomato, big tomatoes, hybrid tomatoes, and hybrid corn, etc. Now let us move on to polyploidy breeding. Polyploidy breeding, many of the plants are vegetatively propagated. They have more than 2 sets of chromosomes, okay? And these chromosomes can be increased by chemicals like colchicine. But when you increase the chromosome number, the seeds become sterile. But these plants cannot be propagated by seeds. They will be propagated by asexual reproduction like cutting. For example, autotetraploid, autopolyploid or autotetraploid means same set of chromosomes is double with a colchicine or something like that. Example, 2n becomes 4x. That's example, potato. Then it can be between a hybrid between 2 or 3 plants. For example, genomes are combined, this is called allopolyploid. Polyploid only, but the source of genes come from different parents. So bread wheat, our bread wheat that we eat is a 6X plant, is a allopolyploid. And we can have a triploid. Many a time when you're eating fruits, you don't want to chew the seeds. You want seedless grapes, seedless watermelon, etc. And these are produced. These are actually triploid. They are produced from crossing a tetraploid with a triploid. And sometimes you see banana is a natural triploid. Then they are there is no seed inside. Maybe vestigial seeds may be there. Okay. Now aneuploid. Sometimes what happens? Some chromosomes may be increased or decreased or anything like that. For example, in grasses which are vegetatively propagated, if you take a grass from one area, it will be chromosome number will be different. In another area, it will be different. Now let us go to mutation breeding. Mutation is what is mutation? Mutation is a sudden heritable change. It's not an environmental change that has happened, but it is a heritable change which has happened in the DNA. Okay, spontaneous mutation frequency is normally very low, that's 10 raised to -6. But we can increase the mutation frequency by subjecting the plant to physical mutagens like X-rays, gamma rays, UV rays, electrons, electron beams, then neutrons, etc. Or you can use chemical mutagens like alkylating agents like EMS, acrin dyes, base analogs, etc. Most of the mutations are recessive. What we get normally are recessive. So in the first generation, it is very difficult to detect them, but the dominant ones also do occur. So most of the mutations are deleterious. Only 0.1% will be beneficial. So if you are getting 100 mutants or 1,000 mutants, only 0.1% will be useful, actually what we want actually. So mutations, induced mutations using radiations can be used for crop improvement, and this is called as mutation breeding. This is a very important method of induction of variation in plants. The seeds are subjected to exposed to gamma rays or X-rays, and then you irradiate them and raise the first generation, go to the second generation, and here you screen for your desirable mutants, and then you carry forward to M3, M4, M5, M6, M7, like that you go, and finally develop a cultivar. So you have a small plant here. After mutation breeding, you may develop a plant with a lot of leaves, if it's something like a lettuce or something like that, where leaf is very important. Okay. So mutation breeding, although mutagenesis is a random phenomena and there's no control over it, several valuable mutants were generated in the past using mutation breeding. 1,500 new varieties of plants were released from using gamma rays. Okay, and mutants for lodging, height, flower color, days to maturity, disease resistant, pathogen resistant plants were released with a mutation breeding by exposing the seeds to gamma rays. And most of these mutations happened only in one or two or more genes, not many genes. But many characters like yield, and they are known as quantitative characters, they are governed by several genes. So it is very unlikely that mutation breeding can be used to induce high yield, but you can develop different mutants and cross them, and by crossing, combining with hybridization, etc., you can improve the yield. Now, I was working in Bhabha Atomic Research Center in Nuclear Agriculture and Biotechnology Division, and we have developed and released 47 new crop varieties for commercial cultivation in different agroclimatic zones in the country. And these are developed with exposing the seeds to cobalt-60 gamma rays. Groundnut, 15 varieties. Some of the groundnuts we developed, they look like a small potatoes. They are the table varieties, 3 varieties of mustard, 1 variety of linseed, then sunflower, 2 varieties of cowpea, urad bean, mung bean, etc. Some of the doshas, idlis, etc. that you are eating in Telangana or wherever you are, they are developed by Bhabha Atomic Research Center, and they are the varieties released by Bhabha Atomic Research Center. Even mung bean and your pigeon pea, your dal, you have a dal along with chapati that is released by, some of the varieties have been released by BRC, then soybean 2 varieties, rice 4 varieties, jute 1 variety, and many varieties have been released in India using mutation breeding. Okay, now some of the recent varieties released in 2018 by our institute are the Trompe cowpea variety TC901. This is a high yielding and 15% increase in yield compared to the national check, and resistant to diseases like cowpea mosaic and root rot diseases and early maturing also. Then another one is mustard, Indian mustard or Brassica gentiana. This has also high yield, and it is resistant to diseases like Alternaria leaf spot and white rust. Now another one is a linseed, which is released in UP, Bihar and Jharkhand. This variety has low linolenic acid and can be used as edible oil. It is resistant moderately to powdery mildew, Alternaria blight, wilt, and resistant to rust diseases. And another variety is a rice variety, which is semi-dwarf with a maturity duration reduced with a lodging resistant, and this also has been released. Mutation breeding. So many mutants have been released. So now with the advent of molecular biology techniques, we can take these mutants and see what happened to the DNA. What happened to the DNA in these mutants? For example, a variety in barley called Mari became very popular. It was released in 1962, And this particular plant was early flowering. It did not lodge, did not fall down. It was short. It is produced high yield, and it was photoperiod insensitive. So then this plant was studied with molecular studies and compared with the Arabidopsis thaliana. You know, Arabidopsis thaliana as a library with a lot of mutants, and the genes were characterized, you know, and one gene called ELF3 gene is characterized in Arabidopsis thaliana. And in our Mari variety, Mari also had a similar modification in the gene, like Arabidopsis thaliana, EF3 gene. Another rice mutant, Calrose, was released in 1976. This is a semi-dwarf mutant with reduced height, decreased internode length, and high yield. This was also studied, and this mutant had a gene called SD1. It is like a green revolution gene, and SD1 coded for gibberellin oxidase. So with the new molecular biology techniques, we can take the mutants obtained and study what exactly has happened. So gamma rays led to base deletions, double strand breaks, and about 400 mutants are released, and 80% of them had a single base mutation. Whereas when you use EMS, that is a chemical mutagen, there is a base substitution. That is how mutation happens. So now let us move on to anthoculture or microspore culture for production of haploids. I said homozygous plants is obtained by inbreeding, selfing for several generations. So, but in using anthoculture, you know what is anthoculture? You know, this is a male part, anther, and it contains the haploid microspores or pollen grains. These can be cultured. Microspores can be cultured, or the whole anther can be cultured in suitable media, and you get haploid plants. And these haploid plants can be diplodized, doubled, the chromosome can be doubled using colchicine. So anthoculture is a technique by which the development of anthers or microspores are cultured at a specific stage in vitro, and you get a callus and embryos or from that. Okay, and the plants are obtained either through embryogenesis or through organogenesis. Microspores can be directly isolated and cultured in petri dishes in liquid media, and each microspore will develop into an embryo. This is a microspore development in a plant. Here is a microspore mother cell or microsporocyte, and meiosis happens. As a result, chromosome number becomes half. Then a second meiosis takes place, where a mitosis takes place and 4 cells are produced. So from a single microsporocyte, you get 4 microspores, and microspores change into pollen grains. So they develop a thick walls around, they undergo division, they produce a vegetative cell and a generative cell, and the generative cell will divide 2 times once and produce 2 sperm cells, and that sperm cells will be used for fertilization. So this is a pollen grain, and this is a microspore. So microspores can be cultured to produce haploid tissue plants, and then which can be diplodized to produce a homozygous diploids or the doubled haploids. The microspores can be isolated from, for example, from Indian mustard and cultured. They undergo typical somatic typical embryogenic stages, and each microspore develop into a typical embryo, and they develop into plants. But all of them are haploids. They can be diplodized by addition of colchicine and pigment. And the culture sometimes gives rise to callus. And the callus can be regenerated into plants by organogenesis. This is a tomato. Haploids have got tremendous applications in agriculture. Gametic number of chromosomes is only half. An application is generation of, as I said earlier, homozygous plants can be generated by chromosome doubling, and the time to produce a variety is reduced by 10 to 6 years using this type of anthoculture, and recessive traits will express very fast. And you can also use embryo culture. Suppose you're crossing a variety of wheat with rye or something like that. What happens? The embryo will be there, but the embryo will not grow. Embryo will develop, but you know the whole seed will abort. When you try to germinate, it will not germinate because it doesn't have the food material stored up. So you can do the embryo culture. So the embryo is isolated from interspecific or intergeneric hybrid, and you can culture them, and you can get an interspecific hybrid in vitro, and it can be used for plant improvement. You can also use somoclonal variation. What is somoclonal variation? Tissue culture, induces mutations. As it is, there will be mutations in the somatic cells, and you can select the somoclonal variation either in vitro or in the field. Suppose you can even combine tissue culture with the physical mutagens like X-rays or chemical mutagens, and you can in the test tube or in the petri dish you can add polyethylene glycol to induce drought tolerance, to select drought for a drought tolerance, or you can add a sodium chloride to select for a salt tolerance, and plants regenerated. Only the regenerated plant, only plants which are tolerant to the particular chemical will be selected and transferred to field. Okay. Now coming to somatic cell hybridization, I already told that plant cells have a cell wall. You can digest the cell wall with enzymes such as cellulase, pectinase, and hemicellulase. The plant cells without cell walls are called protoplasts. Protoplasts can be isolated from different species and from different varieties. I told you earlier that a rice plant cannot be crossed with a wheat plant, but using these protoplast fusion, one can cross a rice protoplast with a wheat plant or with any plant that you want. So one can use a chemical fusion like polyethylene glycol and calcium, or you can apply a fusion electric pulse like a AC pulse when they come together and a DC pulse when they fuse together. So potato plus tomato, we can have a potato tomato. Now let me come to the genetically modified plants. The early 1980s witnessed development of genetically modified plants. So it became possible because of the development of restriction enzymes, because of ligase and other cloning techniques, to clone a gene from any organism, for example, from a bacteria, from a fungi, from an animal, or from a plant, or from a human being, and take out the gene and transfer to plant to produce transgenic plant to improve crop productivity. So as a result, it became possible for scientists, gave an opportunity for scientists to develop a resistance to insect pests and plant pathogens, resistance to high temperature and low temperature, resistance to flooding and drought, enhanced shelf life of fruits and vegetables, high yield, resistant to pollutants and pesticides, and plants with more micronutrients. This is a technique, cloning a gene from a bacteria, putting into a vector, and introducing into a plant and developing the transgenic plant. There are two methods of genetic transformation. All of you would have studied in your DPD classes. There's a agrobacterium mediated gene transfer. There are two bacteria called Agrobacterium tumefaciens and Agrobacterium rhizogens, which are soil bacteria. And these bacteria have got a plasmid, and our gene of interest is introduced into the plasmid, and certain unwanted genes from the plasmid are deleted and introduced into protoplast or into tissues or into meristems, etc., and complete plants regenerated. This is known as transgenic plant. You can take either use agrobacterium as a vector, or you can isolate the DNA, and there's a gene gun called particle gun. This is an instrument where you take out your DNA or the gene of interest, and you take tungsten or gold particles and coat it with the gold particles with your DNA and shoot it out into the tissue. The tissue is kept here, and the gun is inside. You can even have a hand gun, and once a shot is done, you can select the plant for antibiotic resistance or whatever characters you are transferred. Another method is directly you transfer the gene into protoplast by electroporation or by polyethylene glycol calcium mediated, or you can even use a liposome mediated gene transfer. You are all familiar. I'm not going into the details. This is agrobacterium mediated gene transfer and development of transgenic plant. This was developed by Mondego and Jeff Shell in Belgium and later in Germany. Then this is done by John Sanford from USA. The procedure for genetic modification of plants, you can synthetically take a gene from a bacteria, clone it, and clone it into a vector, that is a plasmid, and the plasmid can be introduced into your plant cells, and the plant cells can be cultured, and it will also have an antibiotic resistance gene, which can be selected in vitro, and your plants can be regenerated, taken to the field and test for the gene that you are transferred, whether it's working or not. Then you go to the next generation and see how the gene is transferred to the next generation, whether the transfer is through Mendelian inheritance or a single gene inheritance or whatever it is, you can study. So you all are familiar with, you all have heard about genetically modified cotton for resistance to insect pest, GM cotton. All of you are familiar with that. What is GM cotton? Bacillus thuringiensis is a bacteria which is produces certain crystal proteins, and these crystal proteins are very toxic to several groups of insect pests. So these BT genes, several genes are there, around 200 genes are there. Some of the genes are cloned and introduced into cotton plant to produce transgenic cotton plant resistant to lepidopteran pests. For example, the bollworm. Bollworm is a very serious pest of cotton, and first this transgenic cotton with BT cotton was first approved in USA in 1995 and introduced into India by Monsanto and Myco, and in 2011 India grew GM cotton in 20.6 million hectares, and you see by 2014 95% of cotton in India was GM cotton. Now work done in our lab in BRC, I retired almost 10 years back. This is the work done. Before that, we introduced cloned Cry1AC gene from Bacillus thuringiensis and transferred to some legume plants like moth bean using particle bombardment, and plants developed. And we also modified the Cry1AC gene because the codon usage for a bacteria will be different from for a plant. So we optimized the codon usage of the Cry1AC gene for our plant and introduced the plant to Indian mustard, and the plant was shown to be resistant to diamondback moth. And in another work, we cloned a gene from Trichoderma virens. Trichoderma virens is nothing but a fungus, and this fungus produces is used as a biopesticide. It has a chitinase gene which is very effective, and this chitinase gene from Trichoderma was transferred to tomato for introducing fungal resistance in the tomato plant. In another experiment, another gene called glutathione transferase gene, again from this fungus Trichoderma virens, was transferred to tobacco, and the plant was shown to be tolerant to high temperature, not high temperature, to salt as well as some drought. So this is a transfer of Cry1 synthetic Cry1AC gene. We transferred the gene and it showed enhanced resistance to insect pests. So a synthetic Cry1AC gene was cloned in plant expression binary vector, and the construction was used for a transformation of Indian mustard plants, and their plants were characterized by molecular techniques like and the plant was shown to be resistant to diamondback moth or Plutella xylostella. So this is a different stages in cloning. Cloning in it can be a vector. Then this is a linear representation of with the Cry1AC gene. Then this is a steps in Brassica gensea transformation. Brassica gensea cotyledon with a hypocotyle with a is used for transformation and selected with for hygromycin resistance along with the Cry1AC gene, and then transferred here. Plants showed resistance to diamondback moth, and this gene was the plant was tested for histochemical using the histochemical Gus assay, PCR analysis, southern blot hybridization, RT-PCR, PCR, etc. These are the histochemical Gus assay. Here is a PCR. The control plant is not having a band here, whereas transgenic plants are showing a Cry1AC gene 1.8 kb band. Then insect bioassay is done. This is a control bioassay, whereas transgenic plants are showing more tolerance towards the diamondback moth. This is a stable expression of the Gus gene. You can also use a GFP gene, then a PCR, RT-PCR, southern blot. Another gene that was used is a fungal GST gene for stress tolerance. The plants were found to be stress tolerant when we introduced a when we introduced a GST gene from Trichoderma virus. So this plant showed tolerance to, with a transgenic plant, with a tobacco plant with a GST gene showed a tolerance to polyethylene glycol. That is, it is a drought tolerant. Then also tolerance to sodium chloride. So this is a control plant. This is a transgenic plant. This is a control plant, transgenic plant. So the plant with a GST gene from the fungus was tolerant to sodium chloride, that is salt, as well as drought. Okay. Now we developed a transgenic plant with a chitinase gene from Trichoderma, a biocontrol fungus. So a Trichoderma is here. The tomato plant expressing a fungal chitinase gene showed resistance to fungal attacks. So this work also was done in our laboratory. So you are familiar with the golden rice with increased keratin. Many children in our country, they lack vitamin A, and this source, this golden rice will have a lot of vitamin A, which is like we have to eat otherwise a lot of carrot, which they cannot afford to eat. So golden rice was produced by transgenic technology by other countries. Then this is the first genetically engineered flavor sour tomato. Then we have genetically modified corn, which will increase the farmer income by several million compared to the control, produces enough calories for feeding 88 million people more per year, produces 7.6 higher yield. So top 10 genetically modified foods in the world are shown: corn, soybean, cottonseed, papaya, rice, rapeseed, potatoes, tomatoes, peas, etc. Now molecular markers can be used for crop improvement. So we can score plants at a seedling stage, whether it's tolerant to temperature, whether it's tolerant to drought, etc., by using different techniques like RFLP, restriction fragment length polymorphism, then RAPD, you already know, then AFLP, etc. This is RFLP, which is AFLP and RAPD technique. These techniques can be combined with the plant breeding methods to screen for plants at an early stage to see whether the plants are disease resistant or whether the plants are tolerant to high temperature or flooding or whatever. Okay. Now genomics also has come as a aid in our breeding methods. Genome is a complete set of DNA found in each cell. Genome, the word was coined by German botanist Hans Wingler in 1920. Genomics has come to a long way. So many techniques are there, like DNA sequencing, making libraries, PCR high throughput technology, robotics, computer tools, etc. And I think Dr. Rita Mulekar is going to speak about the genomics. So there is structural genomics, study of the entire genome, functional genomics, proteome, etc. So each crop plants will have millions of nucleotide bases. Arabidopsis has 120 million bases, and information is available for several crop plants. And this information can be incorporated in our breeding programs. Now, latest technique is genome editing in plants. There are several techniques for genome editing, like a protein-dependent DNA cleavage system, like zinc finger nucleases, and transcription activator, like effector talent, ZFN, then CRISPR-Cas9, then RNAi, etc. I'm not going into the details of CRISPR-Cas9 because Rita is going to speak in detail in the afternoon. So CRISPR-Cas9 has been used extensively in plant genetic engineering. This technology has revolutionized agriculture research and engineering. The difference between other earlier technologies and CRISPR is that the CRISPR technology is very precise. You can induce mutation at a specific loci, whereas in the earlier methods like GM crops and mutation breeding, it's a random phenomena. CRISPR-Cas9 act as a molecular scalpels to generate mutations, and it will accelerate plant breeding for improving traits, which will normally span many years. And in a short time, you can improve crops in a specific gene. And not only a single gene, several genes can be manipulated at the same time. For example, in tomato, 6 genes were changed using this technique. So it has major components. I'm not going to the details because Shrita is going to speak in detail about the gDNA and all that. Okay, and double stranded breaks and all that. Okay, using CRISPR-Cas9, corn with high amylopectin, flax with enhanced omega-3 oil, rice, wheat, maize, tomato, potato, soybean with increased yield and mass were produced, plants with a heat tolerance, cold tolerance, resistant to viruses, bacteria and fungal attack were produced, mushrooms resistant to browning. And recently they have even modified the Cas9 into another one, that SRY something like it, to modify that. Yeah. Now let me come to the last part of my talk, smart agriculture or precision agriculture, which is an advanced solution. This is the future of farming, future of breeding crops. It is an emerging concept which will help in management of farms using modern information and communication technologies to increase the quality and quantity of plant products while optimizing human labor. Human labor is going to reduce because people are migrating to the cities, and parents, the children don't want to do farming because they want to have a better job, like they don't want to work in the farms. So one has to switch over to newer techniques for like use of drones, robots, etc. for a smart agriculture. What is smart agriculture? There's going to be a shortage of labor, and you need more food. You have to have automation with the robots, agri-bots you need to have. You have to remove the weeds. You have to spray pesticides, nutrients. Harvesting has to be done, lifting, sorting, etc. will be done by these agri-bots. So many technologies will be combined, like sensors, artificial intelligence, drones, robots connected through internet will gather data and will give you information. It will increase which crops to grow, when to grow, and what is the efficiency in production. It will increase the money the farmer is getting. Heavy farming, you can sit in a lab, sorry, not in a lab, in your house or in a building, and you can control heavy farming vehicles, which can be navigated from the comfort of home through smartphone to perform the task. A GPS will track the positions, and drones equipped with the sensors will gather data, transmit information for analysis. We will monitor the crops for changes in light, humidity, temperature, etc. It will determine the weather patterns in farms. So whole agriculture is going to change in future. Robots will detect and remove weeds. There are robots which can, I know that in IIT Bombay they have developed some robots which can pick up the weeds. Then they can spray pesticide. Wherever there is a disease, they will detect that particular area and they will spray pesticide. They won't spray everywhere. Only in affected plants they will spray pesticides. Then they will analyze the soil, and there is a soil deficiency. They will add nutrients. Then they will check. The robots will check for the acidity, soil drainage, etc. And they will do the work. Then they will detect diseases. Irrigation will be monitored. Then sorting and grading of fruits. Okay, and combine with machine, they will pick up the fruits from the trees, then combine with machine learning. It will use for picking up the fruits, because the images of crops to determine the size, shape, color, etc. For sorting also, image processing will be used. So smart agriculture will become a part of future. Smart farming will increase production efficiency, lower the production risk management, increases business efficiency, higher product quality, and they will kill pests, better pest killers, robotic soil sampling, drone assisted crop monitoring, aerial crop imaging, climate prediction, etc. So it is with less water, with less nutrients, with climate change, you have to produce more using these novel technologies like drones, agri-bots, which can pick up. You don't need labor force. They will go and pick up the weeds. So what is the difference between traditional farming and the smart farming or the precision farming. In traditional farming, century-old practices have been used by farmers, and manual maintenance made. Field data is written down in books, okay, and no way to predict weather, fertilizer, pesticide requirement of the farm. Whereas in smart farming, each farm is analyzed in detail by the data that is obtained, and the computer will suggest which crop to plant, when to harvest, etc. And the computer will tell you which plant is having nutrient deficiency, which area is having a nutrient deficiency, which area is having diseases, and it will suggest remedial methods. And weather also will be predicted for harvesting. And as a result, profit and economy will work out. Now let me come to the future of agriculture. I think this is the last slide. You have to develop climate resilient crops. The novel crops like our hardy crops like pennisetum and ragi and moringa, etc. will be future crops, new crops. Use of novel crops as food source. Then drip irrigation to control water consumption. Like a lot of water is lost by otherwise, so only when it's needed it will be given. And high-tech greenhouses will be grown, like high-tech greenhouses will be grown. Water will be not wasted, it will be recycled, and only biopesticide will be used. Novel biopesticides will be developed. Then biofertilizers like vine fungi and some bacteria which will mobilize phosphorus and other metals into the root system of plants will be developed. And sensors for nutrient control. Sensors will detect whether the soil is having enough nutrients or not. Sensors will be there. And robotics will help in automation. You don't have to spend so much money paying for the laborers. And vertical farming and hydroponics. So when you are, suppose you are working in a company like a Tata Consultancy Services or in Infosys, when you go to the cafeteria, by the side you will see vertical farms. You will see salad plants like lettuce, tomato, etc. growing, and you will be sitting in the middle and eating from the canteen. That's vertical farming, and hydroponics will be there to add to the beauty of the place and also to produce food without contamination from soil and other heavy metals. Thank you.
Thank you, ma'am. Now I request other participants to unmute yourself for the queries.
Good afternoon, ma'am. It was wonderful to listen to you. Such a beautiful presentation on the various strategies of crop improvements and getting more and more yield from the limited space and various technologies. So it was wonderful talk. I think hope participants might have enjoyed, and I hope we will be again getting you back for tomorrow's lecture. So I hope organizers, Sneha madam. Sneha madam.
I have a suggestion for maybe for the next talks. All these participants can write down their queries in the chat box and somebody can read out, so they need not, maybe because it's connected to YouTube also.
Yeah, yeah, we'll be taking these notes, ma'am, surely. And as we are moving ahead time, so lunch, Sneha ma'am, can you announce the lunch actually? We'll have the queries tomorrow's lecture,
sir.
so we can have some more queries from participants.
Okay.
So
Sure, sir. Thank you, ma'am, for nice presentation and fun talking to you. And we'll meet after lunch, sir. If we can give a break for lunch. Thank you.
45 minutes, you announce 45 minutes, and sharply we will be again getting back by 1:45. We will start joining the participants, and we'll tell madam, I mean to say Rita madam, to start at sharp 2 o'clock. Somebody has posted a question about, please ma'am, you note down this question actually, edible vaccines and future something somebody has asked questions, and from our teachers, somebody can note down that question and keep it ready. Sneha ma'am, Rita madam.
Edible vaccines can be produced in banana and other fruits, but you know the dosage is a bit difficult to determine. Even COVID vaccine can be produced in a banana or some fruits like that, but you know how much to eat, one has to study in detail, right?
Thank you, ma'am. As it is lunch time, we take a break for lunch, and sharply we will be again get back. Rita ma'am, if you are listening, please, ma'am, kindly we will start the lecture at 2 o'clock because of the 15-minute, half an hour delay. Kindly note, madam, and
that's fine. I'll be ready at 2 o'clock. I'll join again at 2 o'clock.
Thank you so much. Neha ma'am, is it okay? Shall we leave? Shall we?
Yes, sir.
Shall we
Yes,
end
sir.
the meeting?
Sure, sir. Sharp 2 o'clock we will meet again, sir. Thank you.
No, you start the Zoom at 1:45, and people will start joining, and then ma'am will be starting at that moment. Okay.
Okay, sir.
Ma'am, you now end
We'll
the
do
meeting
that.
from your side.
Yes, sir. We'll do that. Thank you.
Thank you, ma'am. Thank you, Susan, ma'am.
Okay, ladies and gentlemen, let us break for lunch and assemble back by 1:45.
സുബിൻ പോയി പറഞ്ഞായിരുന്നു. ഇല്ല, മറന്നുപോയി.